Greetings, everybody. We have about 65 people now, and given the number who registered, we think we're going to likely have that number rise a little bit. So we're going to sit tight for another minute or two and let more people jump on before we get started.
Oops. Sorry about that.
That's quite all right. Quite all right. All right, we've gotten up to 88. I think it's still rising a little bit. So another minute or two, and then we will be on our way.
Almost 100.
Yes. Oh, good. Excellent turnout. All right, we're at 105 and it seems like it's more or less leveling off. So why don't we go ahead and get started? It's 4:02 p.m. So I think the time is about right. Welcome to the RNA Collaborative Seminar Series. Thank you for attending. My name is Erik Sontheimer and I'll be the moderator today, filling in for RTI Chair Phil Zamor, who couldn't join us today. Before we start, we just want to put in a plug for the Rti Symposium, which we will be holding in June, as you can see on your screen. And we have a fantastic lineup of speakers. And so we just want to encourage all of you to register and attend. It will be held virtually. We hope that we can certainly be closer to getting back to work by then, but we will be virtual this year and hopefully in person again next year, as we much prefer to be. Okay, so with that, it's a distinct pleasure to introduce our two speakers for today, who also happen to be the two newest members of the RTI faculty. They've been tackling the challenge of moving and starting up new labs in the middle of a pandemic, and we are tremendously excited to have them as our newest colleagues. First up today will be Dr. Li Li. After graduating from Fudan University in 2007, he moved to the University of Illinois at Urbana-Champaign for his Ph.D., where he worked with Susan Martinez to discover and understand the first examples of naturally error-prone aminoacyl tRNA synthetases. In 2012, he moved to Jack Shostak's lab at Harvard Med School and MGH as a Life Sciences Research Foundation postdoctoral fellow. And there he pursued a range of studies on the chemical biology of RNA, mostly as it relates to non-enzymatic RNA synthesis, and what we can learn about that from about potential models of prebiotic evolution. The title of Dr. Li's talk today is Efficient Chemical RNA Copying with 2 Aminoimidazole Activated Nucleotides. Dr. Li.
Thank you, Erik, for the nice introduction. So I'm really excited today to tell you about my work in the Shostak lab, where we have developed a new chemical method that can efficiently copy the RNA sequences without using enzymes. And the reason that we got interested into this problem was actually not chemical RNA copying itself. Instead, so the big question that really motivated us was, how did evolution start? What turned rocks into the first living organisms and got this evolution process started and ultimately led to us? And there are two lines of experiments that really influenced our thinking on this problem. The first is the classic Miller-Urey experiment and many new advances in the field of prebiotic chemistry. These experiments collectively showed that we can get these building blocks of a modern life, such as nucleotides, amino acids, fatty acids, from very simple chemical compounds, including hydrogen cyanide, acetylene, ammonia, etc. The second line of discoveries that really laid the kind of intellectual foundation for our work is the discovery that RNA can not only carry the genetic information, but also catalyze biochemical reactions. And they can do this by folding into these very elaborate three-dimensional structures as shown here. Of course, now we know that there are many classes of ribozymes, and in some RNA protein complexes, such as the ribosome, it is also the RNA that plays the catalytic role. So these discoveries led to the hypothesis that an early form of life may contain a membrane that encapsulates these RNA molecules that can replicate themselves. And this much simpler form of life could nevertheless grow, divide, and evolve. And that ultimately led to the current form of life with proteins and DNAs. So if we put these two pieces together, we realize that the biggest remaining puzzle for us to understand how did the evolution start was how can these simple chemical building blocks assemble into an RNA-based self-replicating cell. And this literally is the transition point from chemistry to biology. And if we could demonstrate this experimentally in the lab, that will be extremely helpful for us to understand how could life spontaneously emerge on this planet. But doing so is not trivial. One challenge that has really frustrated the field for a long time is how to use simple chemical method to copy an RNA sequence. So in the rest of my talk, I will show you a new chemical method that we have developed, which can very efficiently copy short RNA sequences that contains all 4 letters in the genetic alphabet. I will tell you how this method was developed, how effective it is, and why it is so effective, and what's its implication for the origin of life problem. And in the end, I will add a little twist just to show you that this method can also be used as a useful chemical biology tool to incorporate modified nucleotides into RNA. So the key molecule in the chemical RNA copying is this phosphoimidazolide. It is a fusion between a nucleoside monophosphate with a 5-membered imidazole ring. And if we compare the structure with let's say GTP, we can immediately see that this imidazole moiety plays the same role as the pyrophosphate leaving group. However, there's a key difference. The phosphorus nitrogen bond is much more labile compared to a phosphorus oxygen bond. This allowed these phosphoimidazolides to react without enzymes. And for this reason, we call them activated monomers. So in a chemical RNA copying reaction, these activated monomers will first bind to the template via the standard Watson-Crick base pairing. and orient themselves to react with the 3' hydroxyl group on the primer to form a new phosphodiester bond. Therefore, the primer is now extended by 1 nucleotide. And this process can be repeated again and again until you get the full-length product. As a proof of principle, Leslie Orgel and his colleagues showed that this kind of chemistry can efficiently copy templates containing only C's and G's. And here's one example showing that for this particular primer, you can get full-length product essentially within a week. However, this kind of chemistry doesn't work so well on the templates containing A's or U's. And as a result, we cannot use it to copy mixed sequence RNA templates. And here's one gel showing you that for this relatively straightforward 7-mer template, the yield of the full-length product was only about 10%. And I want to emphasize here that this modest yield was obtained only after extensive optimization, and it was a big advance at that time. So what else can we do to improve the chemical RNA copy? Would it be possible to develop a more reactive monomer so that we can efficiently copy mixed sequence RNA templates? If you look at the literature, you will find that for more than 30 years, the most effective monomer for RNA copying is this 2-methyl imidazole activated monomer. And the 2-methyl imidazole leaving group was also developed by Leslie Orgel and his colleagues. And because it was so much better than everything else, it has become the gold standard in the field. But what is puzzling is that although these 2-methyl imidazole activated monomers has the highest activity in chemical RNA copying, their intrinsic reactivity is actually not the highest. And here's a plot showing the leaving group pKa versus the hydrolysis rates, which we use as a surrogate to measure the intrinsic reactivity of these monomers. And as you can see, there is a clear linear relationship between the leaving group pKa and the logarithm of the hydrolysis rate. The higher the leaving group pKa, the worse the leaving group. Therefore, the lower the intrinsic reactivity of these monomers and the slower the hydrolysis rate. So that all makes sense. And the 2-methyl imidazole is right here, right? So why don't we use some of these guys with higher intrinsic reactivity for chemical RNA copying? And this is exactly what I did. I synthesized a small library of activated monomers, many of which have a higher intrinsic activity according to our prediction. And then I compared them to the 2-methyl imidazole, which is our reference. Sadly, none of these reacts faster than the 2-methyl imidazole. But the upside is that from these negative results, I learned 2 very important lessons. One is that the smaller the C2 substituent on the leaving group, the faster the chemical RNA copy. And this can be seen by looking at the compounds in the first row. As the size of the C2 substituent decreases from right to left, the reaction rate increases. And the second rule is that a leaving group with a higher pKa will react faster. And this can be seen by looking at the rates from these 5 compounds. So this is actually a very strange result because those monomers with a higher leaving group pKa should have lower intrinsic reactivity, as I have showed you earlier. but somehow they are better at chemical RNA copying, with the 2-methyl-imidazole being the fastest. It took us a while to figure out the mechanism behind this, and I will come back and tell you about this in just a few slides. But at that time, no one said these kind of words to us, and we were left utterly confused. But what I do know is that my experimental data was telling me the ideal leaving group should be both small and have a high pKa. So this led to the design of the 2-amino-imidazole as a new leaving group. First off, this amino group itself is small. In addition, it can form this nice resonance structure, just like the side chain of the arginine. And this increases its pKa to 8.5. And when we make these 2-amino-imidazole activated monomers, they turn out to react much better than the 2-methyl-imidazole activated counterparts. And here's a direct comparison. These two reactions are identical except for the leaving group. For 2-amino-imidazole, you can see that the primer essentially disappeared after 2 hours. And within 3 hours, almost everything has been converted to the +3 and the +4 products. Whereas for reactions using 2-methyl-imidazole, there was still a lot of primer left. And this rate enhancement is independent of the template sequence. For all the templates that I have tested, the 2-amino-imidazole outperforms the 2-methyl-imidazole by a wide margin. And the rate enhancement is most significant for those sequences containing A's or U's. So these results encouraged me to use 2-amino-imidazole based activation chemistry to copy this 7-mer template, which had been a big challenge for us. And it is very satisfying to see that we can now almost quantitatively convert the primer to the full-length product. So, so this is how we discovered the 2-amino-imidazole as a new leaving group and how effective it is. But why is it so effective? So it turned out that the actual mechanism of the chemical RNA copying is a bit more complicated than we initially thought. And this was the work largely done by Travis Wharton, a very talented graduate student, and Wen Zhang, who was a postdoc and now has become an assistant professor in the Indiana University. So what they figured out was that when 2 activated monomers first bound to the template, they would first react to form this very unusual bridged dinucleotide intermediate, where the 2 5' phosphoryl groups are actually covalently bridged by this imidazolium moiety. So this is a highly charged species and is extremely unstable. In most cases, it will be hydrolyzed before it has a chance to react with the primer. What the 2-amino-imidazole does is that it can significantly stabilize this intermediate by delocalizing the positive charge, and at the same time forming 2 intramolecular hydrogen bonds. And this structure also explained the 2 empirical rules that I found from my screen. This C2 substituent has to be small, otherwise it won't fit. And the leaving group should have a higher pKa so that it can better delocalize the positive charge. So the puzzle has now been solved. Since our initial discovery, the 2-amino-imidazole has been widely used in a lot of applications. And this slide summarizes what we have learned from these studies. First, the two amino imidazole RNA copying chemistry has a very good yield. The average single nucleotide incorporation efficiency is 97 to 98%. In addition, this reaction is highly region-specific. We know that RNA has 2 hydroxyl groups. So in theory, we may get a mixture of 3'5' linked and 2'5' linked products. But because of the A-form geometry of the RNA, it turned out that more than 96% of the product has the natural 3'5' linkage. And finally, this RNA copying chemistry works not only for just RNAs, but for many modified nucleotides as well, including those with modified bases and with modified backbones. So what is its implication for our big question? How did evolution start? I think the answer to this question really lies at the interface between chemistry and biology. And here I showed you that by making just a small improvement on the leaving group chemistry, we have solved one big challenge in the field. And it's very tantalizing to think, what if we can further improve the chemistry so that we can copy not just 7 nucleotides, but 20 or 30 nucleotides. This will be very exciting because at this length, RNA can start to encode catalytic functions. Okay, so the last thing I want to share with you is that even though we start this project as sort of like a pure intellectual pursuit, the chemistry that we have developed is actually a useful chemical biology tool for nucleic acid synthesis. So the classic method for nucleic acid synthesis is to use this phosphoramidite. But these phosphoramidite are not very easy to make, especially if you have a special modified base. Because first you have to protect the base, then protect the 5' and the 2' hydroxyl groups. And after the synthesis, you have to deprotect the oligo. And for some modifications, they are simply incompatible with the deprotection chemistry. In contrast, the 2 amino imidazole activation chemistry I showed you is much simpler. From a nucleoside, you can get these activated monomers in just 2 steps. There are no protection or deprotection steps involved. And this whole chemistry works well in aqueous solution. So all of these make it an attractive alternative approach to incorporate modified nucleotides into RNA. Okay, so to conclude, 2 amino imidazole activated nucleotides can efficiently copy mixed sequence RNA templates. And with that, I would like to thank Jack and everybody in the ShortStack lab for making this work possible. When I joined as a graduate, when I was a graduate student, I was trained as a biochemist. And thank you for teaching me all the amazing chemistry. And for my new lab, we will be focusing on the chemical biology of the synthetic mRNA and the development of new synthetic mRNA-based therapeutics. I don't have any kind of preliminary data to show you, but I hope this talk gives you some flavor about the style of the work that we will be doing. So if you know someone who is interested in the synthetic mRNA and have a strong chemistry / biochemistry background, please ask them to contact me. We are hiring at all levels. Thank you, and I'm happy to take questions.
Fantastic. Thank you very much, Lee, for that wonderful talk. I forgot to mention at the beginning that we are asking people to use either the chat function or the Q&A, either one will work, to enter your questions, which I will then read and Dr. Lee will answer. So I'm going to go ahead and start with the first one. In terms of the range of modified bases that are compatible with the 2-aminoimidazole chemistry, you showed some that worked. Were there any that you expected to work, but that were unexpectedly incompatible? Like how wide of a universe of modifications really can be implemented with this particular chemistry?
So that's a great question. So far, it works for actually a wide range of modified nucleotides, which is really surprising. And I think the key is to maintain the two sort of base pairs, because it will form this sort of bridged dinucleotide intermediate. We know that the reactivity depends on the primer as well. And RNA is a very good primer. So in that sense, I think as long as the modified nucleotides can form this bridged dinucleotide intermediate and can bind to the template strongly, the reaction should work.
Yeah. Okay. In the Q&A, Raphael Haslecker asks, did the new leaving group affect the replication fidelity?
That's another fantastic question.
Yes.
From our measurement, it has the same fidelity as the 2-methyl-imidazole activated monomer, which is and of course the error rate depending on which basis you are kind of copying. And the average error rate, I think, is on the order of 1 to 2%. But for specific applications, you can, if you just want to incorporate one specific nucleotide, you can always just use that one nucleotide in your reaction. In our case, because we have to copy all 4 nucleotides, so we are sort of using all of them. But if you only want to use one, then the error rate will not be a problem.
And it's also making me wonder, is it necessary to actually go from the end of the primer in order one by one? Is there any, I guess the binding away from the end of the primer will be disfavored, the stacking will not be as good. So I just wondered if you start forming dinucleotides further out before you actually extend the primer.
Yes, that's another possibility that we don't have much data on like how the sort of dinucleotide will form without the primer nearby. We think that having the primer will help the monomers to bind to it, because otherwise, because the base stacking provides extra binding affinity. But yeah, we haven't really measured like these sort of off-target dinucleotide intermediate formation yet. But
okay. Peter Wang asks, in order for your new chemistry to be used for nucleic acid synthesis, can this chemistry work without a template?
I don't think it can work without the template. Without the template, the efficiency will be very low.
Okay. Peng Yao asks, what is the upper limit of synthesis of RNA in terms of length and fidelity?
So the fidelity is about, so the error rate is about 1 to 2%. The length, so far the biggest we can make is a 7-mer, but that's like a one-part reaction. So we put all the monomers in and put all the helper sequences in. But if we want to use it as a synthesis, what we can do is we can add one monomer, let the reaction to go to completion, wash everything off and add the second one. So it's sort of like a solid phase synthesis. And if we do it in that fashion, I think the actual length that we can copy will be much higher than just 7 nucleotide that I'm showing you here.
Okay. RF asks a good question. Very cool talk. Does an erroneous incorporation at one site affect downstream incorporation?
That's another fantastic question. The answer is yes. People have studied this, and if one mismatch will strongly affect the incorporation of the second position. So, and in many experiments, it has been shown that once there's a mismatch, that becomes sort of like a dead end.
Yeah. Okay. Yeah. Sam Hildebrand asks, have you tried this reaction with any phosphate modifications such as phosphorothioate?
Someone in the Shostak lab is actually working on that, and I think a paper will be published soon. And so I don't want to
okay.
talk too much.
Okay. Give away the game. Okay, we only have 2 more quick questions. Jennifer Miller noticed that on one of the gels where you saw the synthesis of the 7-mer, there were some bands higher up, above the 7-mer. Yeah. Do you know what those are? Yes.
So so in the so that's something that I didn't. So that's some extra details that I didn't have time to sort of fully explain. But in the actual reaction, we have these monomers lined up, and we also have these so-called helper sequences that helps the monomer to bind. These helper sequences, they're also activated. They have the 2 amino imidazole group attached to them, and they can ligate. So some of the extra bands are caused by the ligation of these helper sequences.
Okay. And then the last question from Yerné Mern is, can this chemistry work in a PCR-like manner with exponential amplification?
That's our dream.
Okay.
Yeah.
Maybe if you give it 10,000 years.
Yeah, if we can improve the chemistry to the point that you know, it worked extremely well for a long sequence, and if we can quickly denature the 2 strands and before they re-anneal, then I think we can do it in a PCR fashion.
Okay, so stay tuned someday. Great. All right, thank you very much, Dr. Lee. And in the interest of time, I think we better move ahead. Next up, we have Dr. Wenwen Fang, also a graduate of Fudan University. Dr. Fang moved to Princeton for her Ph.D., where she worked in Laura Landweber's lab to spearhead the discovery of a class of piRNAs and ciliates that play a protective role, not a silencing role, in the amazing genome rearrangements that this organism undergoes during the development of the postmeiotic somatic nucleus. This work earned her the Scaringe Award from the RNA Society in 2013. She then moved to Dave Bartel's lab at the Whitehead Institute as an HHMI Fellow of the Damon Runyon Cancer Research Foundation, where she pioneered high throughput methods to define the basis for microprocessor substrate specificity during microRNA biogenesis. She won a K99 award from the NIH in 2017, and like Dr. Lee, she joined us in the RTI at UMass last year. Her title today is Sensing RNA from microRNA biogenesis to innate immunity. And just as in the previous talk, you should feel free to enter questions into the Q&A. You can do that on the fly, but we will wait until the end of the talk for the actual questions themselves. Dr. Fang.
Okay, great. Can you see my screen well?
Yes.
Yeah, great. Okay, so thank you, Erik, for the introduction, and I would like to thank the RNA Collaborative Seminar Series for the opportunity to present my work, and also for connecting all of us during this pandemic. And I certainly hope that this series will continue after we open up, because it will allow me to attend seminars that I would otherwise miss, for sure. So I'm really interested in RNA recognition in a broadest sense. So I guess to this audience, I don't have to say really how important RNA is. Basically, we see the cell as an RNA machine. And a central question in RNA biology is how are RNAs specifically recognized by either proteins or other RNAs. And I just want to say that cell is an incredibly busy place. And there are a lot of different kinds of RNA with diverse sequence and structures. And so I think when we ask how are specific RNAs recognized, a key aspect of that question is also how are so-called non-specific RNA recognitions avoided. So if we take it apart, cell basically use features to recognize RNA. There is primary sequence motifs. And we now know that a lot of RNA binding proteins recognize very short and degenerate RNA motifs. And I think now it's still a mystery to me how these short motifs will allow RNAs to be specifically recognized. Then we also have secondary structure, tertiary structure, modifications, and chemistries. And I think at the end of the day, the ultimate test of our understanding of RNA recognition is whether we can use these features, a combination of them, to predict the real interactions that are happening in the cell. And I don't think we are there yet. So for today's talk, I'll mostly focus on prime microRNA recognition by the microprocessor complex. And then I'll briefly mention what I also want to study in my own lab, which involves RNA recognition by the innate immune sensors. So briefly, here I just outline the microRNA biogenesis pathway. MicroRNAs are these short regulatory RNAs that repress mRNA expression post-transcriptionally. So they're encoded in the genome and transcribed as a long hairpin-containing transcript called the prime microRNAs. And these prime microRNAs are processed by the microprocessor complex consisting DROSH and DGCR8. Then the pre-microRNA is then cleaved again by dicer, and then one strand is ultimately loaded onto AGO. So I've been focusing on the very initial cleavage step by the microprocessor because this is really the gatekeeper step that restricts which hairpin really becomes the microRNA. And what really defines a microRNA, I think it boils down to the ability of the microprocessor to distinguish bona fide microRNA hairpins from the numerous other hairpins transcribed from the cell. And so very early on, we know microRNA is embedded in hairpin-like structures. And it also requires a lower stem, and so bringing the stem to be about 3 helical turns. In the around 2010, a study from Dave Bartel's lab also looked for sequence motifs in the single-stranded region of the hairpin. And they used large-scale mutagenesis, competitive cleavage, and high-throughput sequencing to find 3 motifs that are in this single-stranded region. And what's left and not interrogated is the stem region due to technical challenges. And so that's where I came in. So I, again, also want to use large-scale mutagenesis and high-throughput analysis to try to find features in the stem region that potentially are recognized by the microprocessor complex. So what I did was to completely randomize these 3 base pair windows in the stem. So here, if I completely randomize this first window in the stem, it gives me 4,096 variants. And I simply walk across the stem and then with one overlapping base pair between the windows, this ultimately gives me about 80,000 variants for this particular microRNA miR16. I also did this for 2 other microRNAs. And in the interest of time, I'm not showing you the actual data, but what came out from this study is this. I'll summarize 2 new findings in this study. One is that there is a surprisingly narrow tolerance for the stem variation. So microprocessor really likes to have a stem length about 35 plus minus 1 base pair. And then it also recognized a complex primary sequence and structure motif called mismatched GHG motif. And subsequent studies from other labs also show that this motif helped position Drosha cleavage to single nucleotide resolution. And what I also found is that none of these features, either primary sequence or secondary structure, none of them are absolutely essential. And in a sense, they are modular. And so we can have a subset of the features and still be able to make a microRNA. And these features can also compensate for each other. For example, if we have a suboptimal secondary structure, then the primary sequence can really rescue that defect and increase the microprocessor recognition. And now with all the features at hand, we can now design microRNA genes de novo. In fact, the artificial microRNAs I can design is processed more efficiently than the natural ones I've assayed with. And so after this study, we really had a very good understanding of what microprocessor is looking for in the substrate. And then I basically went back to the natural microRNAs, and surprisingly there, I see quite a few microRNAs that do not fit to the model, and they seem to have really defective secondary structure. For example, the miR51, it's a very highly expressed microRNA in erythrocyte. It has a very short stem as well as a small loop. The other example, miR181B-1, also has a short stem and yet highly expressed. And so the question then I ask is, how are these naturally occurring defective hairpins recognized and processed? I took a notice that these defective hairpins often reside in the microRNA cluster, meaning there's another hairpin often optimal in their structure, not too far away from them. And so I was wondering, could it be that this neighbor hairpins can somehow help this defective microRNA hairpins? And so I did a rather simple experiment. So here, the miR14451 cluster is specifically expressed in erythrocytes. So here I topically express it in HAC293 cells, and then either on its own or as a cluster. I also use a bidirectional promoter to express miR1 as an internal control in this experiment. And so then I use smRNA Northern to assay for the mature microRNA accumulation. Here, if you express miR144 on its own, you can see a nice accumulation of that microRNA. But if you overexpress miR51 on its own, you don't see a good accumulation. However, if you link them together, you see a huge boost in terms of the expression of miR51. And here I just want to point out you have multiple bands, and this is just because miR51 is a non-canonical microRNA that bypass dicer processing. Instead, it requires exonuclease. And so this pattern is actually expected. And so this enhancing effect is now, we call it cluster assistance. This cluster assistance is a cis effect because if I now move miR144 to a separate transcript, now transcribed together with miR1, I lose this enhancing effect. And so this also rules out the possibility that miR144 is somehow indirectly regulating the expression of miR51. And this cluster assistance tolerates changes in hairpin order, because if you switch the order between the two hairpins, you still see the enhancing effect. But you lose the effect if you substitute the optimal hairpin with a defective one. And so suggesting this effect will require optimal microprocessor substrate. To further test this idea that the cluster resistance relies on microprocessor recognition of the helper hairpin, I then did this experiment. So miR144 stem length is a 34 base pair, which is optimal. I then basically decrease this optimality by shortening the length to 29 base pair. And you can see that miR144 level is decreased, and so did that of miR51. And that can be rescued by compensatory mutations into the 144 to restore the stem length. And also, I can also decrease the optimality by lengthening the stamp of 144 into 39 base pair. And here again, the level of 144 decreased, and the same with MIR51. And that can be rescued as well. So you can really see the level of 51 tracks with that of the 144, which is in turn a function of how optimal it is for microprocessor recognition. And so this really tells us that this process it depends on microprocessor recognition of the helper hairpin. I then explored the spacing between the two hairpins, and in the wild type, it's 95 nucleotide between the two hairpins. And even for up to 900 nucleotide, I can see a robust cluster resistance effect. And so I conclude that this cluster resistance can operate over widely spaced hairpins. So far I've shown you plasmid-based assays, and which assay for mature microRNA accumulation that is a function of its biogenesis and decay. And so to more directly assay for microprocessor cleavage, I then carried out in vitro cleavage assays. So here, I use a lysate that overexpress microprocessor and used in vitro transcribed substrate, MIR51, label at the 3' end, and simply monitor the cleavage over time. And so this is the substrate, and this is the cleavage product that we can see on the gel. And from this, we can derive a rate constant. I can also prepare MIR51 such that it's embedded in this cluster transcript. And so here, you might expect an intermediate where the 144 hairpin is cleaved first. And finally, the product from MIR51 cleavage. So this is exactly what we see. We see full length substrate, intermediate where 144 is cleaved first, and then the 51 cleavage. And so already you can see there is a very obvious increase in terms of its cleavage rate. And in fact, the rate constant increased more than tenfold when it's in the cluster context. And so the big question is how does the cluster resistance work? In the previous in vitro cleavage assay, as I said, I use a lysate that overexpress microprocessor. So the first question is to test, the first thing is to test whether there is additional factor involved in this process. And so here I'm using a purified microprocessor to do the same in vitro cleavage. And as you can see here, you don't really recapitulate the cluster resistance because the rate of 51 cleavage is not higher when, compared to when it's on its own. However, if I add back a lysate that had microprocessor knockout, I can recapitulate cluster resistance. MIR51 is cleaved faster in the cluster context. So this suggests that there is something in the lysate that helps with, that's involved in this process. That's not DROSHA and DGCR8. The fun part of this project really is, back in 2015, I was trying to purify the microprocessor complex towards structural determination. And there I use a step purification where I first pull on DROSHA by affinity, and then also assemble that onto a model prime microRNA substrate, and which has distyl belting that I can pull with. So after this two-step purification, I had a very clean prep. And you can see on this gel, the only protein that's not the known microprocessor component, DROSHA DGCR8, is ERH, or enhancer of rudimentary homolog. It's a 12 kilodalton small protein. And back then, after I found this new component, I was really excited and was trying to figure out what it did. And I really could not find any function, meaning when I knock it down in the cell, I don't see a defect in microRNA processing using what, the standard assay I had at the time. And so now we know cluster resistance. Could it be that this protein is more involved, more specific to this process? And so I literally went back and to dig out the RNAs in the freezer where I had ERH knockdown. So here I use CRISPR interference to knock down ERH. the two separate guide RNAs, DOX-inducible because ERH is an essential gene, and you can see a very good knockdown. And I simply first use qPCR to amplify the precursor across the cleavage site. And so in this assay, if you have good cleavage, you don't see a good accumulation of the precursor. But if you have a very slow cleavage, then you'll see accumulation of the precursor. And so after ERH knockdown, indeed I see MUF1 cleavage is affected. The precursor accumulated to more than 15-fold. In contrast, MUF1 cleavage doesn't seem to be affected substantially. I can also use mRNA sequencing to assay for mature microRNA accumulation, and this is 5 days post-knockdown. I can see MUF1 is the most affected microRNA, and its level decreased about 2.5-fold. And so from this, I conclude that ERH enabled cluster resistance in this cell. And so while we don't know exactly how cluster resistance work yet, but we have some working model that we can work towards. And so I just want to say there are several concurrent studies, and one of them also find another protein called SAFB. It's a nuclear matrix protein that also seems to be involved in cluster resistance. And so the first model we thought about is whether it could be that these additional proteins can mediate dimerization of microprocessors such that binding to one helper hairpin can promote the binding of additional microprocessor to the recipient hairpin. And then the other model is a transfer model where the microprocessor is a monomer, but after cleaving or binding to the first helper hairpin, it can be somehow transferred to the recipient. But we don't know how these additional proteins may be involved in this process. And so we're very interested in working out the exact mechanism. So just to summarize what I told you for the majority of my talk, natural microRNAs may contain defective hairpins, and they won't be cleaved very well by microprocessor. However, it can be cleaved well if it's embedded in a cluster. And this seems to depend on a small protein called ERH that's also part of the microprocessor complex. Because if we knock it down, then we lose the processing of this defective hairpin again. So beyond microRNA biogenesis, I also became interested in RNA sensing innate immunity, partly triggered by the pandemic, I guess. And here the question really is, how does the cell distinguish self and non-self RNA? And I think it's a much more complex problem. It involves many more proteins and pathways. So just to briefly introduce to you, in the cytosol, there are RNA sensors such as MDA5 and Rgi that can detect features such as secondary structure, which is usually present on, say, viral replication intermediate. And that will signal to downstream pathways, eventually leading to the transcription activation of type 1 interferons and pro-inflammatory cytokines. And then there is also endosomal pathway where toll-like receptors can detect endocytosed foreign RNAs, and the same thing it will lead to transcription activation of these genes. And so the question I'm really interested in tackling are what are the specificities of these RNA sensors? How does the cell avoid self-RNA recognition? And are there unknown RNA sensors? Does RNA self-recognition drive autoimmune disease? And so by tackling these questions, I'm hoping that we can provide new insight into not only autoimmune disease, but also virus-host interactions or tumor-immune interactions. And also as we work toward RNA therapeutics, I think it will be interesting to know how delivered RNA talk to the immune system. And so with that, I'd like to thank my postdoctoral lab, Bartel lab at the Whitehead Institute, really everyone there, and also UMass for giving me this opportunity RTI to give me this opportunity to explore what I'm interested in, and also Kate Fitzgerald's lab at UMass for providing their expertise in innate immunity and supporting me in that regard. Yeah, so just want to give a final promotion for our RNA Therapeutics Symposium. I think one unique aspect of this symposium is that you usually have a really good representation from industry, and I learned a lot from them, and I hope that you will feel the same. So yeah, that's it.
All right, thank you very much, Wenwen. That was terrific. And we are opening it up now for questions. I'm going to start. I hope I didn't just miss this, but if you take a well-processed microRNA, but that is not the native miR-144, which is, if you just swap out a good one for a different good one, but that is different, do you still get the cluster assistance?
Yes. Yes, okay. There is an experiment where I swap out miR-144 with miR-125, which is another natural microRNA, has optimal structure, well processed. It works also. So it doesn't have to be 144. Yeah,
the reason I asked is because I was wondering if there's any possibility you must have looked at this for, loop-loop interactions or, something that would be specific between those two that would augment the structure in some way.
Yeah, so far I don't have any evidence for that. It doesn't have to be 144, but could it be that 125 also have some loop interactions? I cannot rule that out. Sure. But so far it seems to be that any good hairpin would work.
Okay. Jiping Wang it says great talk, and then also asks how many microRNA hairpins need a helper?
That's a good question. I think, there are several ways of testing this. I haven't done it myself. There are several ways of testing this. One is actually, you can basically use, synthetic method to make microRNA individually and put them into cluster and see whether being in the cluster help with their accumulation. And that can be done. And the other way is you can knock down or knock out, I guess you cannot knock out ERH, but you can knock down ERH or the SAFB and to see which microRNA levels are affected. Presumably, those that are affected are those that are that require cluster assistant to be processed well. Although that's a little bit indirect. So actually, the SAFB knockdown has been done, maybe ERH as well, maybe in Nari's paper. I haven't looked at it, and it's also cell type specific. And so because some microRNAs express ubiquitously and some are restricted to certain tissues and cell types. And so if you do this knockdown study in cell types, it will be cell type dependent, which ones require cluster assistance. But I know about 40% of microRNAs resides in clusters, and some has multiple members, not just two. It could be 6, and the largest one I think it's 41. And so it will be interesting to find out the scope of cluster assistance. Yeah.
Okay. We've racked up about 8 different questions now, so let's see how many of them we can get through. Jiping followed up with, can you predict those that require cluster assistance from their sequences? I think you said at the beginning that these tend to be suboptimal for the parameters that you defined earlier, right?
Yeah, so that's something that we've always wanted to do, first of all, to predict microRNAs, right? From a genome or transcriptome, can you predict microRNAs? And the hurdle, the barrier there was to predict secondary structures, because we know that the optimal length is 35, but when you really fold using the whatever software we have, how do we derive that parameter, knowing it's 35 or how long where does the stem end? So that I'm not an expert in bioinformatics, so I'm really interested in knowing the answer, but I haven't done it myself. So if we can predict, or if we can try to predict microRNA, we will know which will require clusters, because basically those are the ones that are defective. That we can have a score for all the hairpins, and those have below a certain threshold may require clusters to be processed.
Okay. Henry Chang asks about the biology. He says, great presentation, Wenwen. Do you have any idea why certain microRNAs are designed to be coupled like this? Are these microRNAs used for particular functions?
Mm-hmm. You know, I tend to think this is a neutral thing, that because you already have a good neighbor, you don't have to evolve a good structure, you know, you can be expressed. But then, you know, I talked to, you know, I was interviewing, so I talked to a lot of people, quite a few men was proposing that maybe it's a regulatory function. Usually if you have something that that's not optimal, maybe cell want to have a lower expression at some point and a high expression at some point, and you can toggle this, you know, if you have additional factors that's responsible for this. And so I was wondering, what if we sort of fix MIR51 in mice or a cell, so that it's optimal, it's always expressed optimally. And is that a problem for the cell to have that? I don't know. I initially just thought it's neutral, that it can be processed really well in this way, so just stayed. But it's also possible that it's a regulatory mechanism to regulate microRNA biogenesis. Yeah.
Okay, yeah, and so along those lines, we have a question from Ali Saleh. Thanks for the great talk. So could the defective loop, I think he means like the functional loop or connection, be responsible for the variance in the expression level between mirror cluster members? So if you have multiple microRNAs that are made in a 1-to-1 to 1-to-1 ratio as a precursor, but they don't accumulate at that same ratio, is that variance in the ultimate expression level partly because of this processing effect that you're seeing?
Well, if anything, this cluster resistance will dampen the difference between different microRNA hairpins, right? Because 144.1 should be very different. Now they are kind of similar. And so yeah, that would be my explanation, that so clusters will help those that are not optimal. And in terms of the final expression level, you know, it's a function of both transcription, processing, and then loading and decay. It's a multi-step process. Okay.
I'd love to get to the remaining 5 or so, but we're already 3 minutes over and we want to be respectful of people's time. So we'll just close out with one last question from Junji Guo. Is the helper effect stoichiometric? In other words, what happens if there are multiple poor hairpins near a single good one?
Multiple weak ones with a single good one?
Yeah.
Yeah. So there's only one experiment I did in this, as for this. So I linked 144 to 51, but also to 181B-1, right? The one good one with 2 bad ones. And I saw both weak, the bad ones increase, and they seems to be kind of competitive with each. So the level of increase doesn't seem to be as much as when they are linked just 1-to-1. But that was just the only experiment I did in vitro assay. So does it help, you know, understand the mechanism? I'm thinking about this.
Yeah.
But yeah, if you link one good one to 2 bad ones, both will be helped. That's my in vitro study. What that told me.
Okay.
Yeah.
All right. I took a screenshot of the remaining questions that we weren't able to get to, so you can look at them later. I'll send it to you. In the meantime, we're already several minutes over, so I want to thank everybody for sticking with us all the way through to the end. And I especially want to thank Dr. Li and Dr. Fang for a couple of terrific seminars. Thank you very much. And finally, I just want to thank everybody for attending. It was great to have such great numbers. And I think that will do it for today. So thanks, everybody. And we'll look forward to seeing you again at the next RNA Collaborative Symposium. Thank you.